Hi, this is Pallavi Raman and I will be explaining my project developed as part of the course EEL 6825 Pattern Recognition. The title of my project is Motif Discovery in Biological Networks and I will be explaining in detail the various components of my project. Coming to the overview of this presentation, first I will introduce the topic and define the terms used in the project. Then I will shed some light on how the input was obtained and how it is processed. After input processing, I will explain the computational methods used in this project. Afterwards, I will give a small demo or simulation of this project and the results will be discussed, and finally the project will be summarized. So, what are motifs and why motif detection is necessary? Biological networks provide great potential to understand how cells function. Network motifs are key structures through which biological networks operate. Identifying motifs in biological networks has great potential to uncover important network functions and characteristics. Studies have been done on the relationship between the functional behavior of a motif and its structural topology and also the overrepresentation in a particular gene network. The frequency of occurrence of a network motif can shed light on very important phenomenon, and the overabundance implies that the genes have undergone some phenomenon which has led to this overabundance. This phenomenon can be a mutation, which can be a form of a disease or any other secondary condition. Hence, the ability to determine the motifs computationally is an essential step in pursuing these research efforts. Well, the complicated problem of finding the motifs in a network of gene sequences can be mapped to, graph theory problem. It is same as identifying subgraphs containing keynotes which are overrepresented in a given graph in a network or graph G, which is same as finding n letters long pattern which appears exactly in every sequence given a set of sequences. Hence, graph-based algorithms are used for obtaining the overrepresentation of a subgraph in a network of graphs. We deal with two types of networks in this project. The probabilistic networks have probability values on their edges. A deterministic network is one with no probability occurrence values on its edges. Well, biological networks undergo mutation from time to time, so we cannot say a protein interaction or a gene interaction occurs every time. So there ideally in real time there should be some probability value associated with each edge. So we'll be dealing with probabilistic networks in this project. Coming to the applications, the discovery of network motifs is not just limited to computational biology but has applications in other networks such as electronic circuits and power distribution networks, the software engineering diagram, the internet, World Wide Web, social networks, and even the ecological networks like the food web. Now that we have seen the importance of motif detection and its applications, let's see how the input is obtained and how it is processed. In this project, I have worked on gene sequence information obtained from KEGG pathway database and string interactions information obtained from the string database tool. Well, let's see what roles this KEGG database and string database tool have played in input processing. KEGG offers a pathway database which is a core of KEGG resource. Well, this is a collection of databases dealing with genomes, diseases, drugs, biological pathways, and chemical substances. So this pathway database is a wiring diagram database and is a collection of pathway maps integrating many entities including genes, proteins, chemical compounds, RNAs, disease genes, and chemical reactions. The classification of this pathway maps are genetic information processing, metabolism, cellular processes, genetic information processing, human diseases, and drug development. Well, the pathway map used in this project is of the carbohydrate metabolism. The process is called the glycolysis or gluconeogenesis. Glycolysis, in simple terms, is the process of converting glucose into pyruvate and generating small amounts of energy. Among the databases and resources available for getting protein-protein interactions, String, the search tool for retrieval of interacting genes or proteins, is an excellent database. Well, this database provides the network graph using the pathway maps obtained from KEGG. So hence, this is the network we have obtained from the String database. This shows the protein-protein interactions between two proteins or the genes. Hence, the nodes are the molecules and the edges are the protein-protein interactions. So we obtain a pathway like gene information, gene sequence information from KEGG database, which is input to String database, and String database formed a network from those interactions, and this is the input we give to our project for further analysis. Coming to the computational method, the complicated problem of finding the motifs in a network of gene sequence can be mapped to the graph theory as explained in the earlier slides. Let's see this more in detail. The graph form of a biological network can be represented with each node representing a molecule and each edge representing interaction between two such nodes. Well, using this graph, an adjacency matrix is formed with P cross V entries. V is the number of nodes, and the entries are filled with either ones or zeros depending on whether an edge is present or not. Well, using this adjacency matrix and a corresponding edge probability matrix which contains the probability values related to each edges, we calculate two frequency definitions. Well, there are three frequency definitions, but only two will be calculated in this project. The third can be taken up as future work. Well, the first frequency measure F1, it deals with overlapping instances and it has no constraints regarding to overlap. So it counts all occurrences of a motif regardless of overlapping of edges or nodes or both. Well, the second frequency measure F2 deals with the number of occurrences of a motif so that no two occurrences share an edge. That means it just allows only a node overlap. Hence, F1 covers the overlapping frequencies and F2 covers the non-overlapping frequencies. We have used various graph algorithms and we calculate the overlap values for each subgraph or the motif. So let's discuss how this is done. Well, before analyzing the steps, let me revise some terms related to graph theory. A subgraph is a graph whose set of vertices and edges is a subset of the set of vertices and edges of the main graph. And for a given motif, all non-identical subgraphs in G in a graph which are isomorphic to the motif are called the set of embeddings. So this embeddings is a set of all non-identical subgraphs which are isomorphic to the motif. We deal with such a set of embeddings in our project. Well, given an input probabilistic graph with genes, proteins as nodes and protein-protein interactions as edges with an edge probabilistic matrix containing the probability values on the edges in the input graph, the first step is to find all embeddings of a motif in a corresponding deterministic graph. So each probabilistic graph can be considered as a set of deterministic graphs. As we can see in this figure, this is the probabilistic graph which is given, which is taken as an input, and This is one of the deterministic graph which can be obtained from this probabilistic graph. The second step is to create an overlap graph for a set of embeddings of this motif. As you can see, this is the motif pattern. This is actually the pattern 1. We are considering the triangle pattern, and this is the overlap graph corresponding to this motif pattern. The third step is to calculate the existence probability value. The existence probability value, which is nothing but it is the product of the probability of edges of the embedding pattern, and and also the neighbor overlap value. This neighbor overlap value is a probability distribution. Is a probability distribution because the neighbors the neighbors to a particular node can vary depending on the probability of the interaction between these two nodes. Hence, this forms a distribution, and we calculate the expected value of this distribution. And the third step is repeated for all patterns in the set of embeddings obtained in the first step. The third step is calculating the existence probability and the neighbor overlap probability. The final step is to pick the node with the highest overlap value. This overlap value can be obtained by just dividing existence probability by the expected value of the neighbor overlap probability. So in the set of embeddings we pick the embedding with the highest overlap value, and we remove that embedding, and also all the nodes, all its neighbors which are overlapping with this embedding. This step is repeated until the overlap graph which we constructed in the step 2 becomes empty. So the output frequency F2, which is obtained by adding the existence probabilities of the embeddings. This is the output which is produced in our project. Well, let me give a small demo of my project. We are obtaining these F1 and F2 values of 4 different types of patterns. The P0, P1, P0, P1, P2, P3, which are nothing but the angle, the triangle, chain, and star patterns, which are shown here. So this is the cake pathway database, and this is for the glycolysis, gluconeogenesis, and I am considering only for Homo sapiens, the human beings, and this is the gene information. This is the input which needs to be processed further to be used in our project. So the, this is the gene information which is obtained from the kick pathway database, which from this I have just obtained the names of the proteins and their interactions. So using this regular expression, I can just filter the nodes which I give as input to the string database tool. Well, this is the network which I obtained after inputting the gene sequence information from the kick pathway database. It has different data settings. Well, I have just considered co-expression and co-occurrence, as the earlier network was much complex, and there are options for exporting this data, this network as an Excel file. So well, this is the interactions file which I have obtained from the string database tool. So this is the node 1, node 2. This means that there is an edge between these two nodes, and we do have this co-expression and co-occurrence values and the final probability values which we will be used in our probabilistic network. Well, coming to the execution, this is the graph, this is the data. This is basically a graph information with a set of edges and the probability corresponding to these edges. So I have given the motif size as 3, and let's see what the output will be. As you can see, the number of nodes are 67 and the size of the motif is 3. So it will calculate, so it has calculated the F1 measure for pattern 0, which is an angle. That means there are 329 overlapping sequences and there are 24 non-overlapping sequences. Let's see for the motif size 4. I have used lesser number of nodes for calculating the size for patterns as with the increase in the number of nodes, the time complexity increases. So for fast execution, I have used lesser number of nodes. So the F1 measure for the pattern 2 and the pattern 3 are given as below, and the F2 measures are also calculated. So the pattern 2 is the chain pattern and the pattern 3 is the star pattern. Well, the results, as I have shown, the motive of this project is to find the non-overlapping and overlapping frequencies given a probabilistic network graph in polynomial time complexity. So we have achieved that. Coming to the conclusion, this method uses F1 and F2 measures for motive frequency counting and is focused on independent motive instances in probabilistic networks. F3 measure is a much complicated frequency measure for calculating the occurrences of the motives. Very few studies have concentrated on this measure. The current method can be extended to calculate F3 measure in the future. Graph theory algorithms are always open to optimizations. The important criterion here is producing the results not only accurately but also within a polynomial time duration. The future work can be focused on finding the F3 measure, extending the application to other motifs differing in shapes and sizes, and also optimizing the running time of the code so that runtime is polynomial in terms of input.